So pretty much this program is made by students and by professors, and they are here to select you during the afternoon. And please feel free to come and talk to them during the seminar afterwards, and then to ask questions. So I'd like to introduce John Godleski. John Godleski had his MD at the University of Pittsburgh. He's an associate professor in the Department of Environmental Health at HSPH, Harvard School of Public Health, the school that our program has a collaboration with. And he's also an associate professor of pathology at Harvard Medical School. And he's the founder of our program. So the program started some years ago when he received Paula Saldiva there. So when you go there, you'll be received by him and by his wife there. So please welcome John Godleski.
Well, thank you all for coming, and especially that you're coming at a time that is your vacation. So I appreciate very much that you're all here during this time when you really should be out on the beach and enjoying all sorts of things. When I realized to come here and to arrange this program is a lot of work. And I also know that as we've gone around, we've seen everybody wearing Brazilian soccer shirts and other shirts of their team. So for the hard work that people have been doing, I've brought some shirts from Boston. And this is the Boston Red Sox, the baseball team. And the Boston Red Sox won the championship last year of baseball. And so I have some shirts for the people who organized this meeting for us. And first is Rudolfo, who's done a great job in coordinating the alumni. And thank you very much. The second shirt, and I have these in order so they're the right sizes, is for Professor Burns, who had, again, a Red Sox shirt, so he's ready for his trip to Boston. And hopefully this fits him. Please. And
Burns with the Red Sox is deserves of it.
And finally, for Professor Saldiva, who has been the backbone and support of this program, he is also for the Red Sox as well. And when he was in Boston, his son thought that maybe he would not be a Red Sox player, but he would be in management of Red Sox. Yes. So this is all important, and as we look at things in Boston and things in Brazil, the intensity for sport is almost the same, if you can believe it. It's really a pleasure to be here to talk to you and tell you a little bit about the program. And so as I start off, I'm going to tell you first about the exchange program and give you some facts about it over the years, and tell you what it is, and then we'll talk a little bit about my research. And let's see, up this way. So the Brazilian programs at the School of Public Health, and there are several. There's one in population health and infectious disease with Marcia Castro, Mary Wilson, John David, and this is with Santa Casa Medical School. So this is not the only program in the School of Public Health. There also is our program in environmental health, that we coordinate and what we're here to talk about today, and you'll learn more about. And one of the interesting things is that I am now the faculty coordinator of all Brazilian programs at Harvard School of Public Health. And so we're really looking for not only to continue this program as a very strong entity, but also to build new programs and consider new opportunities for Brazilian students and faculty and people advancing their careers in Brazil. So this is a very important advancement that has happened at the School of Public Health. So our program, where you spend 1 year doing research in the laboratory, and the term is January to December. Now, why 1 year? 1 year is important. In 1 year, you can learn your way around the lab. You can really start thinking about what you're doing. You can learn some techniques, and you can begin to apply those techniques in an orderly way that really gives you a real feeling of what science is about. People who come and stand and watch and see what's going on in a laboratory really get a very small amount from it. Getting involved, getting engaged is really the important part of it, and that's what the unique part of this program is, that you come in, you get your hands dirty, you get into it. This is important, and this is what we want to foster, and this is the kind of programs we want to develop, such that we're really allowing people to have their own success. The students have a research tutorial class with me, you attend seminars, but most importantly, you complete a laboratory project with a faculty mentor. Current faculty members, and there's a whole list of them, most of you who've been looking at the program know as much about these people as anybody, and the laboratories that they coordinate. The current distribution of students is about 1 to 3 per lab. So in some labs there's 1 student, in some others there's as many as 3. The funding of this program over the years. The program began with Professor Saldiva's coming to Boston for a year. But then when we started to really take students on a regular basis was in 2005. So you will be the class of 2015. That's an important 10-year milestone for this program. So I see it as a big deal in terms of the people who will be selected for this year. In 2005, we had 3 students who were able to have the means to take a year off and study abroad, and then the number started to increase after those 3 students. In 2008, just 3 years later, we had 10 students, but all of them didn't have the resources. So we started to develop funding through the Lehman Foundation, to Tender Bank, and the other thing that happened at this time is the Harvard David Rockefeller Center office in Brazil opened with Jason Diet, who helped to develop a lot of this funding in coordination with people here at the school. The students themselves worked toward this. So this was all a very important step, and these 10 students of 2008 were very significant because this is the first year where we had outside funding. From 2009 to 2010, we were fully supported by philanthropy, and really this program is a model for Science Without Borders. What kind of things could be accomplished by students spending a year abroad? In 2013, the students were supported directly by Science Without Borders, and the philanthropic support was promised if needed. And in 2014 and beyond, the support will be from Science Without Borders, from Brazil-Harvard agreement, and by philanthropy to some extent as it's needed. So these are all critical features of the funding that allows not only students from Brazil with means, but also students of Brazil who come from backgrounds where they don't have the means, that are allowed to really participate fully and take advantage of this program. A very important feature. So what have we accomplished? 2015 is the 10th year. And by this time, we'll have more than 80 alumni in the program. We have 40 publications, 8 first author publications from students in the program, and many more in the, as we say, in the pipeline, papers to come. The students have done multiple poster presentations at local, national, and international meetings. Hilario Francelino, who was here who was in Boston 2 years ago, presented a medical poster at a meeting in Russia, and he won top prize based on the research that he did. So it's really possible that you can not only expand your horizons to the United States, but essentially all over the world. This is very important. Students write reports on their total experience at 6 months. Generally, they tell us how cold it is when you come in December or in January. But also, they give us some real insight on the program. A final scientific report is written at the end of the year by each student. And this is really in the form of a paper. Some of those papers are ready to send off to journals for publication. Others are good scientific reports with data that will probably be used in other publications. So, and then all of the students present their work at our department seminar series, usually in December, and this is always a big time both for our program and for the students, because they have to stand up and give a 10-minute presentation to the program. We have really what I would call a vibrant, active alumni program here in Brazil, connected to the David Rockefeller Center. They've had research seminars, networking with students. They've done Skype meetings that many of you were there when I gave you a presentation a few weeks ago. And there are many return visits to Boston and meetings with Harvard faculty visit Brazil. I think Lucas has been back at least once that I know to work on the data that he generated while he was there. So these are all important features of the program. Now, the focus of our collaboration in my lab is air pollution. It's a major problem in the cities of the world. Living in Sao Paulo, you're fully aware of this. As we look at pollution, in the United States, the big pollution event occurred in 1948, a long time ago, in Donora, Pennsylvania, when an inversion created very high levels of pollution where people actually were seen to die related to that pollution. This was in 1948, and it was really the event in the United States that catalyzed the interest in pollution control. And interestingly, in the United States, the fact that this happened in 1948 took 15 years before the United States government actually put in place funding of a program to look at air pollution. It took another 7 years before legislation was passed that actually began to control air pollution. So that in the United States, at least since 1970, we've had both laws and a will to reduce air pollution. We've done this somewhat successfully, but at the same time, around the world, that's not happening. In the past year in China, we've heard about what they called air apocalypses. And what this was is that pollution was so bad, as high as 1,000 micrograms per cubic meter. And actually they said it was 1,000 milligrams, but that's not possible. It was really 1,000 micrograms. But this was reported in the New York Times just last October. And we've heard about similar ones in Shanghai, in Beijing, and other cities in China. So that it's really been a problem. At the same time, we look at Sao Paulo. And on many days, as you look over the buildings, you see this visible layer of brown haze over the building. This is a high level of air pollution. When I was here 2 years ago, it was 100 micrograms per cubic meter. As we flew in on the airplane, we could just see the brown haze. This kind of thing is something that Brazilians need to work on controlling, and you need to work on as you begin to understand this, as you become advocates for control of these kinds of events. Clearly, this is not a healthy situation. Epidemiologic studies, toxicologic studies of air pollution have been done for a number of years. Most epidemiologic studies in the United States have air pollution ranges between 5 and 30 micrograms per cubic meter, and yet they still find effects. Average levels in the northeastern United States are 11 micrograms per cubic meter. We did toxicologic studies a few years ago at 300 micrograms per cubic meter. And the idea that we did them at that level seemed extremely high. Yet we look at what's going on in China today and other parts of the world, in India. Levels like this have for exposing the general population. But interestingly, we are now doing toxicologic studies using specifically sources of traffic-related air pollution. And we're doing these at levels ranging from 30 to 50 micrograms per cubic meter. 30 to 50 micrograms, that's half of what the pollution level is sometimes here in Sao Paulo. So if we can do toxicologic studies and find significant findings, you can only imagine the effect that this has on the pollution of the people, or on the health of the people living here. As we look around the world, we can see that contributes. The green in here is traffic, and the blue is also traffic. So more than half in most places are coming from traffic sources. If we think about traffic sources, traffic causes pollution to come from the tailpipe, from the roads, from other sources. But there are also things that happen once the pollution gets into the air. And so we think about what comes from the tailpipes of cars and buses and trucks, what comes from the tires and the brakes as primary, but then what happens in the air to the gases as secondary. And what happens is that once these are emitted directly into the atmosphere, these gases and particles react with the sun, and we get a photochemical reaction of the primary pollutants to form secondary pollutants. The mechanism of secondary particle formation includes nucleation, condensation, coagulation, so you get almost molecules formed in the air as a result of these interactions. These molecules coalesce to form particles. The particles are what ends up affecting the lung and the cardiovascular system and the neurologic systems of the body. As we think about these secondary pollutants, most of the studies have used gas chromatography to differentiate the primary and secondary components. The fraction of secondary pollution varies anywhere from 39 to 90% depending on the location, and they're formed primarily from nitric oxide precursors, and that can vary from 4 to 37%. Why is this important? Secondary organic aerosols comprise a large portion of PM2.5. Gaseous precursors and secondary organic aerosols include volatile organics such as benzene, toluene, ethylene, what comes from vaporization of gas and diesel fuel, and nitrogen oxides. The sources, formation, composition, spatial temporal variability are not entirely well understood, and the potential differences in toxicity between secondary and primary pollutants is one of our areas of interest. Most studies have really only focused on the primary pollutants, but we're looking at both primary and secondary. Now, what about trends in PM2.5? PM2.5 is the particles that you can see, that brown haze over the city. And as we look at these in the United States, they are ranging in 2001 to just below 14 on average, and now it's down around 12. In this 10-year period, there's actually been a 24% decrease in pollution in these annual trends. Remember what I showed you in terms of other locations, and the fact that these are in this range, and yet we can still see health effects with this kind of average is very important. In the United States, we look at carbon dioxide. Carbon dioxide is a very important marker of what's happening in the air. And as you can see, carbon dioxide was going up. Now it's somewhat leveled off over the past few years. If we look at Sao Paulo in terms of averages of pollution, you can see you're around 25 micrograms per cubic meter. And importantly, it's not really decreasing. This is important, and this is something that really must be at the uppermost of people's concern. Because looking at the difference between Sao Paulo in this 7-year period versus the United States in that period, the trends are different. And this is important. Now, if we look at other parts of the world, we look at Europe. They started to come down in 2004 to 2007, but now they're actually going up, whether you look at urban traffic, just traffic sources, or rural. And to some extent, this has been based on what's been included, but the fact is that the pollution levels in Europe are still relatively high. If we look at CO, and CO measured in Hawaii, which is one of the major measuring sites for carbon dioxide in the world, you can see that there's this constant upward trend. Now, as we look at that, we have to think about where the pollution is coming from that contributes to that trend. If we look at wind directions in the northern hemisphere, in the United States, our direction is mostly west to east, so that, and we know that pollution that starts in the Midwest affects the East Coast, but then it clears off the coast And whether it eventually gets to Europe, it probably does, and probably contributes some to that. We know that you can measure effects and components that you would find only in some part of the world and different parts of the world. If we look at South America and Mexico, Mexico City being one of the most polluted, the pollution problems in Sao Paulo and the other megacities of South America. These are all going toward this Hawaiian measuring station, and also they're going down towards the poles where pollution is also measured. And if we look at China, China is actually contributing both to what's going up to the North Pole as well as some of which comes across the United States. So these wind patterns become very important, and it reminds us that we live in one world, and how we look at pollution in our own backyard not only affects us, but affects our neighbors, and not only our neighbors in a neighboring country, but the other side of the world as well. So you really need to consider this truly a global perspective as you work on this. And when we think about carbon dioxide, you have to think about the balance. The production of carbon dioxide from all sources versus the consumption of carbon dioxide by the world's plants. And this is a very simple thing that you learned in grade school about how photosynthesis converts carbon dioxide to oxygen. But as we look at that, what's happening is the production of carbon dioxide from all sources around the world is increasing, and the consumption is decreasing. Now, how do we know that it's decreasing? Here's a very interesting map of the world. And this was done by satellite at the University of Maryland. And what they were able to do was map over a 10-year period the loss of forest. And so that as you look at areas like Canada, Alaska, and North America have lost a lot of forest. You can look at down here at Brazil and what forests have been lost in Brazil by clear cutting. Going across, looking at Europe, there hasn't been much of a change, but look at all the forest deforestation in China. So that as we look around the world and add all this up, it becomes staggering. Almost a million square miles, 2.3 million square kilometers of forest have been lost in 10 years. This is incredible as we think about how we need to worry about CO2, both from the perspective of generation of CO2, but also from the consumption side. So these are all important considerations as you think about pollution and where we're going. Now, what about our research? Well, we've been focusing on vehicular emissions. Epidemiologic studies have shown respiratory outcomes, including increases in asthma, bronchitis, COPD, as well as lung cancer. Most of the epidemiologic studies look at how they relate to whether people live near roads, near major roads, or in more rural areas. Cardiovascular outcomes include ischemic heart disease, circulatory disease, coronary artery disease, myocardial infarction. A number of these have all been identified. In epidemiologic studies, they use the residents near roads or black carbons or other markers of traffic. Laboratory experiments have been done with collected emissions, usually from diesel vehicles. You can look at a single vehicle on a dynamometer so that you control how the vehicle runs. And we've been more involved in field experiments. One approach by some people has been to move a particle concentrator to near road locations. Our approach has been to go to a source, study both primary and secondary particles separately and together. So how do we do this? Well, what we're trying to do is we're trying to just look at this vehicle source. And in order to do that, you then have to eliminate all the other potential sources of pollution and just focus on the vehicle. How do we do that? We use the mixed vehicular emissions that can be collected from the ventilatory system of a tunnel. We use a tunnel that rarely has traffic jams, has a mixture of vehicles representative of urban traffic, and has nearly the same flow of traffic each day. Reproducibility. So it's a primary destination tunnel. It's the tunnel that goes to the airport. Same taxi cabs go back and forth. Same number of cars. Same number of trucks. But very important that then we can study these in a very reproducible way. Our hypotheses are that mixed vehicular particulates collected from this tunnel will have cardiopulmonary toxicity. The primary particles, secondary particles, and mixtures will have all different cardiopulmonary toxic responses. So here's what the tunnel looks like. It has 45,000 vehicles per day going through. It has fairly high levels of PM10, similar levels of PM2.5, again in the range of 30 to 60. How does this work? As we look at this picture, you can see the car going through the tunnel, and all these open spaces is where the ventilation goes out. The ventilation goes out from and we have a photochemical chamber in there. We have the vehicle emissions that we can irradiate to model sunlight, and we produce an organic aerosol, and we run it through a denuder to remove gases. And then this goes directly to a toxicology laboratory for animal exposures. So here's the schematic of this, and we can either filter out the particles before they come in or not turn the lights on to get different outcomes. This is a photochemical chamber. It then goes to this denuder system and then to the rats. This is what that photochemical chamber looks like. It's 8 cubic meters. It's quite large. These are all the lights around it to mimic sunlight. This is the denuder that takes away the gases. And as we look at this, the particles and gases go in this direction, and clean air goes in the other direction, and by diffusion it goes out and lowers the concentration of gases in the exposure air. And so this is the study design that we use, where we're looking at different scenarios of primary particles, primary plus secondary, secondary alone, primary plus gases, secondary plus gases. And these are where we expose the animals in these individual plethysmographs. They're on telemeter receivers so that we can measure cardiovascular parameters. This is our mobile lab. Here's a telemeter. And here is the rat in the exposure chamber. The rat is smart enough to sort of duck his head away from the flow of pollution. When we look at the exposure, and here's one paper that was done, you'll recognize some names on here of people that have been from University of Sao Paulo, Vicente, Helena, Rodrigo Sato in this case, and what we're looking at, you can see here's our actual measurement of concentration. It's in the range of 50 micrograms per cubic meter. And if we look at respiratory flow in the animals, as we're able to measure in this plethysmograph, you see that the animals receiving the pollution exposure have decreased airflow as they breathe. These are the control animals receiving filtered air, and on each day it's lower in the control group. If we look at bronchoalveolar lavage, we wash out the lung and look at the cells. What we see is that there is an increase in inflammatory cells with the exposures, and as we look at this, we see that all three exposures produced a decrease in inspiratory and expiratory airflows, tidal volumes and minute volumes on bronchoalveolar lavage. Secondary particles had an increase in neutrophils. Primary particles had an increase in lymphocytes, and the combination of the two had increases in both. If we look at cardiovascular parameters, again, a study where the concentration was in the range of 50 micrograms per cubic meter. The control data shows that the two groups had essentially the same response to filtered air. But when we start looking at the different exposure scenarios, here's primary particles, here's secondary, here's primary plus secondary, and you can see that the effect of this is an increase in systolic pressure, an increase in diastolic pressure, as much as greater than 10 millimeters of mercury. Here in the combination, it's approaching 10 millimeters of mercury. It's a highly significant change in both the parameters. So that as we look at the blood pressure findings, we had a sustained elevation higher than 10 millimeters of mercury, higher than previously observed. The secondary particles in the first 2 days had an elevation, and then it actually went down after a few days. So there was a different pattern with this, and with the combination, the net effects were similar to the two similarly, where they started out high, and then by the third week it was no longer significant. So in summary, using an urban tunnel as a source of traffic emissions, we can produce aerosols for animal exposures composed of primary traffic-derived particles alone, secondary or combinations. Primary particles alone, secondary particles and combinations all affect the breathing pattern, airflow, and blood pressure substantially. And primary particles alone increase pulmonary lymphocytes, secondary particles increase neutrophils, and the combination increased both. Primary particles had sustained increases in blood pressure after the first week. Secondary particles significantly decreased blood pressure at the end, and the combination had increases for the first 2 weeks. So homeostatic mechanisms to control increases in blood pressure, limit and reverse the amount of change during longer and multiple exposures are important. Increases in control of blood pressure are very complex processes, involve many control systems, and this is some of what we continue to study. As we look at the future directions of where we're going, we want to understand both the unique and common features of air pollution in different parts of the world. We're studying the toxicologic effects of different formulations of gasoline and alcohol. We're studying the health effects of air pollution from different sources of air pollution worldwide, and we're trying to understand the biologic mechanisms responsible for the observed health effects. Here acknowledges a lot of the people from USP that have been in our program that work specifically with me. You can see a lot of familiar names up here that have been involved in our studies. Here's a picture of Rodrigo Sato. Rodrigo actually has had 4 publications out of the studies that we've done. Here's a picture of Fernanda and Hilario with my wife Mary Lou, which unfortunately you're not getting a chance to meet on this trip. And here's a picture of Rodrigo and Andresa at a fair painting some children's faces. And usually when students show up in January, they eventually see snow. And here's a picture of my backyard after a snowfall. The students a few years ago got a chance to meet Drew Faust, the president of Harvard, and you can see many of your colleagues in this picture, and the opportunities that they've had. So I thank you very much for your attention, and I'll be glad to answer any questions that you might have during the break, or if we have some time now. And thank you very much.
So, if you have any questions, please.
Thank you for this presentation. And considering those data about the increasing speeding of particulate pollution in the States compared to all around the world, what do you think is driving this? Is it technological improvement of engines or different qualities of fuel, of diesel or gasoline? We know that we have differences in this quality and in this technology in Brazil, for example, compared to the States.
Well, I think
both are involved, but what do you think is the most important?
Well, I think there are several things. First of all, the laws are an impetus for sure. The pollution laws in the United States have become stricter and more strict, and those are the impetus. The second is the inventions, and the inventions of industry, of the automobile industry, to really produce cars, produce combustion systems that really can produce essentially carbon dioxide and water as their products. And so those are important. The third is the will of the people. That people really want to see clean air. They want to have clean air, and so that people will spend the extra money to buy a car that has less pollution, to buy a hybrid, to buy an electric vehicle, to add solar electricity to their houses, and so forth. So there's a lot of reasons for the decrease, but I think it's important that the data shows that when there is a will of the people to make this decrease, and there's both convincing evidence from science and that will, that you can really begin to show these kinds of decreases. And so I think it's important both to train people of how this can be accomplished, to train people to do the studies and to show the health effects that make the case for the need, that people will believe and understand that there's a need to do this. So these are all features that I think have been the basis of that decrease in the United States. And the important consideration is that human studies, epidemiologic studies, even at these levels, continue to show that there's health effects. And my laboratory studies done at 30 or 50 micrograms per cubic meter show a health effect. And 10 millimeters of mercury of increase in blood pressure is a significant health effect. And so you can't ignore that sort of thing. So these are the kinds of things that have gone a long way to both reduce pollution in the United States and really show a need for worldwide pollution decrease. The headlines are on the basis of the extraordinary high levels, but we really need to decrease things day to day so that it's better, it's cleaner, it's a place where we can live more healthy lives. And this is very important for physicians from the perspective of preventive medicine. And this is what we're trying to do. Not for just one person, but for the populations of the world.
So do you have questions for the students? Feel free to ask now or to ask during the coffee. So I have a question about, you mentioned that we have a role and we explain better in terms of science and having the concept of bringing to change, but how to bridge the gap between basic science and knowing that we need to do this and to change, and to have a practical action in terms of public health policy?
Well, I think it starts at levels where we are right here. If you can convince the young people of the world that this is something that's very important, that needs to be done, this starts then an attitude in a whole generation that becomes very important. And I think it's this kind of convincing. The other thing is that we need to not just talk to each other. For example, in the past year, I've given talks at Boston College Business School. I've given talks to community groups. And Professor Saldiva, I understand, is on television a lot. All of these things are very important, that we can't just talk to other scientists. We have to talk to the public and make it clear that these are important issues. And as we do this, I think the message will become clear. And I think to some extent, at least in the United States, the news tends to sensationalize things. I think and people get tired of hearing the sensational story, because it doesn't make sense to them, because one day you hear one thing is bad for you, the next day you hear something else is bad for you, and maybe that's not good. So you can't do that. You really got to have a consistent message that comes across clearly, makes the point, and can convince people, not just scientists, but the general population. And I think this is very important.
Don't wait, just the last question about, anyone has another question now? So, usually now is about a program, so most of the students are going to interviews today, and then some of them will pass the program, some are not passing. So I'd like to ask about what is your advice to them in terms of what you're looking for, just to be sure they know what you, professors, are looking for, and also to the students who are not being selected this year to apply again or to search for another program. What is your advice about this?
Well, I think one of the big advantages of our program is that it's established, that people know what to expect from the students when they come in, and know how to train the students to be as successful as they can be. So that's a big important point in thinking about coming to our program versus others. The second thing is, I think in those students who are selected for this year, I think that will be wonderful. But for those students that aren't selected, consider applying again. Because I've seen in the experience that we've had over the last several years, people who are not selected in one year often ask me for advice on what they should do to improve, and I try to give them very individual advice for what their shortcomings are. And once they do that and they apply again, they're often the top candidates. And so that, for example, I have a student in my lab this year who's doing a fantastic job. And that 1 year of additional schooling made a big difference. So that I think you get to this point where you have interviews, you've accomplished a lot. If you make it, that's wonderful. If you don't make it, I think you should continue to work toward it. I think there are things that you can do. You can shortcut the system and try and find another place. Whether that place will be as organized and ready to receive you as our program with its history versus others is a decision that you would have to make. But you can do this in any number of years. We've had people do it anywhere from the 2nd to the 6th year. And all of them are equally successful. So I think we're looking for students that have a real desire to do the program, have an interest in science, have an understanding of how to become a physician scientist in terms of how you look at patients, how you treat patients, how you look at the data that comes to you as a physician and interpret it and understand it. All of these things are things that you get out of this program. You learn individual techniques, but most importantly, you learn a way of thinking and a way of approaching experiments and problems and data. And all of this is something that you can use the rest of your life. So that we're looking for people that are well organized, that are able to take on responsibility, because when you come, you have a lot of responsibility. Over the years, I've seen students come and they do everything with their cell phone, talking to their mother to get approval to be able to do this, whether it's to rent an apartment or to do an experiment in the lab. And I've seen students come and take on a responsibility that is given to them, learn how to do it, practice to do better, and then do things very successfully. So there's a maturing process that happens in this program, and all of those things are what we're looking for is students who have the potential to benefit from this program the most. And although you apply this year, you may not be judged to have the potential for this year, but you very well may have the potential for great success in the next year or the year after that. So I would strongly encourage people that if being part of this program is your dream, if you're not selected, to stay with it and come back again for another round. And I think some of the people, as I've looked through the applications, I remember talking to some of those people last year and interviewing them last year, and I look forward to seeing how they've changed in a year. And it's my bet that every one of them will have come a long way in their maturity, in their knowledge, in their interest and understanding of who they are and what they want to do. So I'm looking forward to talking to all of you, and we should have a fun afternoon and tomorrow.
Thank you again. Mr. Godleski.